to 9 antibiotics out of the 17
of DNA, which is the sulfonic group, would improve the efficacy of the dispersants. So our results indicate that the introduction of conformational mobile states, in this case of an butyl group between a plain aromatic residue, which is formed by pi interaction with the graphene or reduced graphene oxide surface, and a polar head group in our case was sulfonyl
fluoxacin and enrofloxacin. The antimicrobial resistant profile of each E. coli strain was clustered using a graphical or heat map representation using color to indicate the level of resistance. The left side clusters were shown the antimicrobial resistance profile against antibiotic, and these have been seen that they clustered according to their culture media, mainly according to their culture media, which they have been isolated from. On the top, we can see the cluster of the antimicrobial agent according to their pattern of resistant strains. The table here shows the variant type or the gene sequence type for the genes, the beta-lactamase genes that have been identified. For the CTX type genes, the most predominant type belonged to the CTXM32. Also, there was TEM1 and TEM30 have been detected for the OXA, BLA-OXA genes. There was the BLA-OXA1 type of beta-lactamase. For the whole genome sequencing, the serotypes and the sequence types of the selected strains shown that these types were both reported before from animal sources and in humans. So same E. coli sequence carrying the same sequence type and the serotype have been isolated before in both humans and animals. Here I'm going to show the genomic environment for some of the beta-lactamases gene, and if they've been carried by transponsible element or inserted in a chromosome. So first, starting with the beta-lactamase CTXM14, A, which is present in two of the sequence strain, BCC2 and BCE3. The genes were found to be carried by transponsible element. It found downstream of insertion sequence ISECP1 and upstream of the another insertion sequence, which is IS903. This transposition unit have been reported before, but the location of this transposition unit inserted in a chromosome, which is inserted in a chromosomal gene FDEC1, haven't been reported before. This figure shows the sequence alignment comparison between the fourth sequence strain using Artemis comparison tool. The genomic environment of this gene showed that between BCE3 and BCC2 there was a similarity because both of the BLATEM 1B was carried by a TN2 transposon, just that the variation was in the direction of the genes because it present in a reverse orientation. The gene BLATEM30 and BS7 was carried by TN3 transposon, and the gene of BLATEM1A, which was carried by A2.5, was carried by a tourniqueted TN3 transposon, which was completely different from the genetic environment comparing with the other three strains. The sequence alignment comparison between the BS7 and A2.5 BLA-OXA1 region using Artemis comparison tool showed that there was 100% similarity between these two strains regarding the BLA-OXA1 region. The gene was carried on integron class 2, and which was located with the other element of TN21 transposon, with other antibiotic resistance genes such as AADA1, which confer resistance to streptomycin, and with the SAL1, which confer resistance to the sulfonoamide resistant sulfonoamide antibiotics. These clusters of antibiotic resistance and two strains, which have been isolated 1.5 years apart of each other, can indicate might indicate the ongoing circulation of such resistant cluster within the farm environment. The conclusion that can be derived from this study that there was multi-drug resistant ESBL producing E. coli isolated from the farm environment. There was a diversity of the genotypic of these strain and their relation to their phenotypic of antimicrobial resistance, which indicate the presence of clonal and non-clonal relationship between those different strains. This might indicate the presence of these genes on transponsible element, which could be enable the separate of such genes in the environment via the horizontal gene transfer. Using whole genome sequencing allow us to understand the different antimicrobial resistant phenotypes of different strains, the presence of 4 strains with the same with an sequence type, which has been reported previously in both humans and animals, might indicate the success of these strains in surviving in different environments. So here I go to the end of my presentation, and I will be happy to answer any question. Thank you.
Okay, thank you very much, Dr. Delvin, about your presentation. Please now we can move to the question sessions or part. And we will start with the Professor Dr. Vanessa Molkand, and her presentation was about the Sri Lanka's tea waste in the fashion opportunity with significant increase in the value added exports of Sri Lanka. Any question you have, you can ask Dr. Vanessa. Thank you.
I would like to thank all the speakers, and I'm open for any questions. I would also like to thank the other moderator, Mr. Kais Kader, who is attached as a scientific researcher carrying out his PhD in animal physiology at the University of The Hague. And he has joined the University of The Hague after completing his MSc in animal physiology from Ukraine. And I would like to thank all the presenters, Dr. Rajni, Dr. Hashim, Dr. Delvin, for wonderful presentations. And I'm ready for any questions you all may have. If you don't have any questions from my presentations, the session is open for any questions you have for any other presenters. Dr. Rajni, I have a question for you.
Yeah,
Dr. Rajni, do in India people consume this plant, or how much is, or is it only in Banaras, or it's in all over India that people consume?
Ma'am, this plant is actually, the Partolica oleracea plant is consumed by basically tribal and local communities, not only in Varanasi, but it is consumed in many parts of India, like Andhra Pradesh, Arunachal Pradesh, Bihar, Chhattisgarh, Gujarat, Goa, Mizoram, Tamil Nadu, Rajasthan. As I told you that its distribution is cosmopolitan, so it is broadly distributed. But just because of lack of knowledge, and there is a gap of difference of a knowledge between generation to generation. This plant is now going to extinct. So we have to again rediscover this plant and use as a plant for meeting our UN SDGs.
Okay, thank you, Dr. Rajni.
Thank you, ma'am. Thank you so much.
Any other questions from the other participants? Okay, while we are thinking of questions, can everybody switch on your webcams so that the organizers can take a picture of us, the whole group? Everybody, can you please switch on your webcams?
On.
Dr. Hashim.
Dr. Hashim, are you there? You have some questions you may answer.
Yeah.
Yes, Prof. Vanessa, maybe you say your questions to Dr. Hashim. He's available now.
Yeah, the session is open. Anybody who wants to ask questions from anyone, including me or any other presenters, the session is open for question and answers. So if we don't have any questions from the sessions, shall we have a normal chat for about 5-10 minutes on the challenges faced by each one of us by doing the research what we have been doing in our respective countries. So shall we start from Dr. Delveen?
Yeah, thank you for you, Dr. Vinitha. Actually, I performed that work in another country. I did it in UK as a part of my PhD research. It is hard to do the same work here in my country due to the limited facilities that is provided and the funding as well. So if the funding is provided, we can do the same work. Actually, because the subject was very important, there is underestimated for the using of antimicrobial in animal husbandry and their effects on human health as a source, being as a source of the transferring of antimicrobial resistance genes that cause infection also in human and in animals. So if there is facility provided, funding provided, we can do the same work here in our country.
Dr. Delveen, like how much time do you think, like according to your experience, the facilities what you obtained in UK, how much time do you think, in how many years, like do you think that the same facilities could be obtained in Iran?
It is not hard, you know, you just need people to understand that you need it to work. So in the UK, when you work, you have, you know, your lab, you have everything provided for you. Here, if you do any steps, you need to like transfer between one place to another place to do each step. So it's going to take long time until you finish the project. Because each of the instruments supplement is provided in another lab. That lab is, there are many people they work on it, so you have to wait on the queue to do it.
Yeah, I think we all face the same condition irrespective of the
And the
country
Middle
where
East,
we
you
are.
all
Yeah.
have that problem, and it is not hard to do it. Just they provide like one lab with all the equipment. It will be easy.
Yeah. See, I'm going to give you an example of the country. Like in Sri Lanka, we do have like the lab conditions were not so good. So all of our students who were doing their PhDs went to USA or UK to complete their PhDs. And so we still have a huge problem of brain drain because people who go maybe about 10% return and 90% stay over. So government did open a huge research organization like StraighTech, and it's a public-private partnership. So like the government doesn't have to pay throughout or maintain that throughout. So what they did was they funded 50% of the whole system, and they asked private funding from the 50%. So I think that's a good example which other countries could also take. Because we almost always rely on government funding, and then we tend to do basic research. So, and then what happens is developing countries like Sri Lanka, which has many more demands than only research. So what happens is government can't afford to cope up with expenses. So it's always a good practice to include a public-private partnership in a research organization. So then we will be doing the industrial relevant research, right? And the fruits of that research would be reaped by the normal community much more faster than 100%, basic research government research organization. So that's a trial. We have had many falls, but still we are in that process ongoing. So just wanted to share that experience with you all. Dr. Hashim, we would like to know your experience. Have you done your complete stay, or have you just wanted to know how has your research field been, and what is your experience of developing and developed world? Have you worked in a developed world? Dr. Hashim, you would like to share your experience with us? Okay, so I'm sorry I can't hear Dr. Hashim. Can anybody else hear Dr. Hashim? Okay, in the meantime,
Dr. Hashim, the question is for you.
okay, till the meantime, Rajni, do you like to share your experience of working in India, and have you had international exposure, and how do you compare with Indian institute where you are working?
Yeah, thank you, ma'am. And first, I would like to thank you, Vinitha, ma'am, for your nice presentation. And it is a very good idea to convert tea waste into the fashion opportunity. So first, I wish to convey my regard towards you for this nice presentation. And of course, ma'am, luckily I am in a Banaras Hindu University, and Banaras Hindu University in terms of facility, we have a lot of facility in comparison to other institute in India. But of course, there are lots of schools scope of improvement in India. And as compared to the other countries' laboratory, there are a lot of scope of improvement, and little bit gender biasness is an issue in India. So in spite of male faculty members and many other, female got less opportunity, especially in the if she want to work in a field condition. So this type of challenges we females are facing in developing countries like India. And as an ecologist, basically I'm an ecologist. So my work is mostly on field-based, forest-based, and laboratory-based. So day to day, I am facing these type of challenges. Expert manpower is very these are some problems I wish to share you. Thank you, ma'am.
Dr. Rajni, I think your connection is, we can't hear you properly.
Okay, I think there is a network issue.
Right.
Hello.
Yeah,
There is
now
a
we can hear you back.
Issue, ma'am. That's why.
Yes, now we can hear you.
Yeah. Ma'am, okay. Hello, I think there is a network issue, ma'am.
Yeah, Rajni, if you turn off your video, I think we should be able to hear you fine.
Yeah, ma'am. Because there is a little bit network issue. So, ma'am, I like, personally, I like your presentation very much, Vinitha, ma'am. And it is a very good idea to convert tea waste into a fashion opportunity. And I wish to stole your idea to converting other type of waste into the fashion opportunities, because there are many type of wastes generated in India, like molasses and many other things. So I will talk you later. So how to convert these things also into the product, means convert these things into productive way. And the second thing is, okay, ma'am, I think there is a network issue, so we will discuss later, I think. Frequently, I am losing my connection.
Right. Anybody else who would like to share your experience? Thank you so much, Rajni, and you're doing a great work, and hope you will continue doing it.
Thank you so much.
And we have met in a virtual contact, and hope we can meet in person too.
Personal, okay.
Anybody else would like to have some questions or share their experience? So in absence of any questions or comments, let's ask the organizer to take a group photo. So can you please, everybody, put on your cameras again? Amal? Professor Amal, can you hear me?
Yes, I hear you, Vanita. Yes, good morning. Hello.
Yeah.
Thanks for accepting my invitation. Dr. Shaheen.
Pleasure.
Dr. Shaheen.
Yes, doctor, I'm with you.
Can you take photo?
Yeah, sure.
Yeah. Can you take photo?
Yeah.
Yeah. Okay. Thanks.
Why don't you turn on your video as well?
Yeah. Okay. Thanks for accepting my invitation, all of you. Thanks for this nice session and for your experiences at the end. I think you have to wrap up.
Thank you so much.
Thanks a lot.
Thank you for the wonderful event.
Thanks a lot for your presence
Stay
and your
blessed,
lecture.
blessed.
Yeah, thanks. I hope to meet you again after that long time. Definitely.
Yes, definitely.
Yeah, soon.
Thank you, Delvin. Thank you, Rajni. Thank you, Hashim. And thank you, Kayas, for wonderful work. Thank you so much.
Bye-bye. Please join us in another session now about science communication. Bye-bye.
Sure.
Okay.
Bye.